Okay, so to have a better view of this video, I would advise that you put your phone in a landscape mode. Yes, so that you can see everything clearly as it is done. Now, as you can see on your screen, 2025 WACE Biology Specimen is out and the exams is to commence in the next few days. So how prepared are you? That is why your favorite teacher, Glory Jonathan, is always here to put you through and to guide you through your study. So all what I want you to do for me is to get your writing material, fix your eyes to the screen, and then try to absorb everything I want to show you in this video. And if you can do that, I bet you will come back here to say thank you to your favorite teacher. Okay, let's. Specimen A is a bean weevil. It belongs to the kingdom Animalia, phylum Arthropoda, class Insecta, order Coleoptera, family Chrysomelidae, genus Acanthoceles, species Optectus. Specimen A, bean weevil, has complete metamorphosis. That is, they lay eggs. From eggs, they change to larva. From larva to pupa. From pupa to adult stage, otherwise called the imago. Bean weevil are parasites. They are crop pests. Now, what are the possible questions you should be expecting? You will see questions like, identify the class of Specimen A. So, the class of Specimen A is Insecta. Then the next question should be, state reasons for your answer in number 1 AI. That's state reason why you say that Specimen A belongs to the class Insecta. So here, you should mention any observable characteristics you see. So, for example, what are the observable characteristics? Bean weevil have segmented bodies. As you can see, they have rigid exoskeleton. They have exoskeleton, they do not have endoskeleton. They have jointed appendages or jointed legs. Their bodies are divided into 3 segments, that is the head, the thorax, and the abdomen, or you can say they are triploblastic. Now, the next question you will ask is, what are their economic importance? Now, the economic importance of Specimen A, when we talked about economic importance, it has to do with advantages and disadvantages. So, see, because they say importance, you are not supposed to say only the important. You should combine both the advantage and disadvantages. Both the advantages and disadvantages. So, what are the economic importance? They destroy bean grains through feeding or breeding. They contaminate stored grains with dead bodies, remains, or feces. They reduce the economic value of cereals. Application of pesticide or insecticide in its control is expensive. Reduce viability of invested grains. It also reduces farmers' income. It reduces market value of grains. So any of those ones can go for the economic importance of Specimen A. Now, another thing you may be asked of methods of controlling specimen A. So, what are those methods of controlling specimen A? You discard infested food in outdoor trash. Freeze suspect food at 0° Fahrenheit for 6 days. Clean cabinets or shelves where infested food are stored with soap and water. Stored all dry foods in a glass or plastic container with a tight lid. Proper drying of seed to reduce moisture, and also store grain over fireplace. All of these are the methods of prevention or control of bean weevil. Now, they may also ask you mention types of crops that bean weevil attack. So it's not because it is called bean weevil, it means it attacks only beans. So other types of crops it attacks are rice, maize, with bean inclusive. B is a bean seed with holes. You may be asked to identify specimen B. So do not just write specimen B is a beans, but write specimen B is a bean seed with holes. Yes, because you can see that it has holes, so make sure you put holes. Specimen B is a bean seed with holes. Now the next question should be, give reason for your answer. Why you say specimen B is a bean seed or is a seed, that's the key word. So you are going to look for observable characteristics of a seed. So the reason why you answer that specimen B is a seed is that specimen B has a seed coat or a tester that protects the embryo or the seed. Specimen B is a seed because it has helium, which serves as point of attachment of the seed to the seed stalk. It also has a microfill, which permits air or water into the embryo. Specimen B also have an embryo which germinates. So all these four make specimen B, all these four characteristics make specimen B a seed. Now, the next question should be, what are the observable features of specimen B? From what you have seen, specimen B has a curve shape or a kidney shape. It has a scar which shows points of attachment of the seed to the seed stalk. It is white. You can also write it is brown. You know, some beans are black. You can write it so like that in color. Now it has holes. You should also fix that. Now, what type of seed is specimen B? Specimen B is a non-endospermic seed. Why do we say so? Because food is stored in the cotyledon and not in the endosperm. That makes it a non-endospermic seed. Now, specimen B has, the kind of placentation it has is marginal placentation. Okay, so now the follow-up question you should expect is, mention conditions necessary for specimen B to germinate. So those conditions necessary for seeds to germinate are water or moisture, warmth or suitable temperature, air or oxygen, viable seeds. So the reason I came up with these conditions necessary for germination is that bean seed with small holes can often still germinate, especially if the holes are not extensive or have not damaged the embryo. So they may likely ask, what is the condition that will make specimen B to germinate? Okay, now they can ask you, what kind of germination does specimen B have? Specimen B have apigeal germination. And apigeal germination has the following characteristics or features. Testa will first split, then hypocots will emerge uncovered. Cotyledon are carried above the soil. Energy obtained from food is stored in the cotyledon. Hypocotyl elongates. Cotyledon is the first photosynthetic tissue. Yes, so those are the questions you should expect to see. And then, what's the agricultural classification of specimen B? They are legumes. And what is the class of food it provides? It provides protein and mineral salt, example, iron. Now, specimen B is from the kingdom Plantae. It has a phylum, Magnilophyta. That means it is a flowering plant. Magnilophyta means that that plant is a flowering plant. It belongs to the class Magnoliopsida or dicotyledonous plant. It belongs to the subclass Rosidae. It belongs to the order Fabaceae. It belongs to the phylum Fabaceae. It belongs to the genus Phaseolus. So, specimen C is a water leaf plant. You may be asked to identify the class to which specimen C belongs. Specimen C belongs to the class Magnoliopsida or dicotyledonous plants. Then you may be asked to give reason why you say specimen C belongs to dicotyledonous plants or Magnoliopsida. So what you do is to remember the characteristics of a dicotyledonous plant which you had learned. So I'm going to mention them out and you are going to write any one you remember in case that kind of question should come up to identify the reason why specimen C is a dicotyledonous plant. So those characteristics of dicotyledonous plants are: it has two seed leaves or cotyledon, it has taproot system as you can see in the diagram, it has apigeal germination, it has net venation, there is presence of xylem, it undergoes secondary growth, the floral part exists in groups of 4 or 5, vascular bundles are arranged in regular patterns. Now, you may be asked to give observable features of specimen C. So, in observable features you are going to write exactly what you see. So, what are the observable features of water leaf? It has broad and spatulate leaf. It has taproot system. It has knit or reticulate venation. It has succulent stem or succulent leaf. It has petiole. It has bright colored flowers, or you can say bright flowers or colored flowers. There is absence of haze. There is presence of branches. Yes. And you may be asked to name the functions of some of the parts you have mentioned. Like we have the functions of the root are: it anchors the plant to the ground, it absorbs water and mineral salt used by the plant, it stores food for the plants. The functions of the stem or the functions of the branches are: they hold the leaves in an upright position to receive rays of the sun. It conducts water or mineral salts from roots to leaves. It conducts manufactured food from the leaves to the roots, and it holds the flowers in a suitable position for pollination. Now, the functions of the leaves is that it aids in food production by photosynthesis. It aids in transpiration or transpiration pool. It admits energy. Now, what type of shape does the leaf of specimen C have? It has obovate. The shape of the leaf is obovate. And I've said earlier, the venation is net venation or reticulate venation. Okay. Now, the kingdom of specimen C is plantae. The division is Magnoliophyta, which means flowering plant. The class is Magnoliosida, as I said earlier, which means it is a dicotyledonous plant. The order is Caryophyllales. The family is Thalinaceae. The genus is Thalinum. The species is Fructicusum. Thalinus fructicusum. Some people call it Thalinus triangula. So you may be asked to make a transverse or a cross section of the roots of specimen B, of the root of specimen C, or the stem of specimen C. And thereafter you may be asked to view it with a hand lens, because it will be provided by your teacher. And then after viewing, you will be asked to draw what you see. So you need to be conversant with drawing. So if you have not watched my video on the common mistakes made in biology practicals, please search for that video and watch. Just go to my video content and search for it. It is there. Then you try as much as possible to avoid those mistakes as you made your drawing. So looking at the screen, you can see the roots of a dicotyledonous stem, the roots of a dicotyledonous plant, and the stem of a dicot plant. So try now to acquaint yourself by try now to know how to draw these diagrams. But in this my diagram, there are some parts which are missing. Yes, the parts are actually there, but they are not labeled because my screen is too small to accommodate the drawing. Parts like epidermal hairs, it is there, but it is not labeled. Pitch or parenchyma is there, but is not labeled. We have collenchyma, it is there, but is not labeled. So get your textbook, study the diagram, and label all parts, because as I said in my earlier video concerning your exams, my video on common mistakes made in practical biology, you do not know the parts the examiner is looking for. So make sure you get your textbook and study the diagram very well so that you can label all parts and not lose your marks. Specimen D is Quill of domestic fowl. The kingdom is Animalia. The phylum is Chordata. The class is Aves. Order is Galliformes. Family is Phasianidae. Genus is Gallus. Species is Gallus domesticus. So in case you are asked to state the class to which specimen D belongs, the class is Aves, and you are asked to state the reason why you say specimen D belongs to the class Aves, you already know what to do. Give the characteristics possessed by Aves class, as we have done in our subsequent specimen. Now you may be asked to state the function of specimen D to the organism that bears it, or yes, the function of specimen D to the domestic fowl. So the functions are flight, that is, it is used in flying. It keeps the birds warm. If you don't want to say it keeps the bird warm, you can say it maintains constant body temperature. It is used in camouflage. It is used during sexual display. It is waterproofing. That is, it protects the bird from getting wet. Now, you may also ask to give the location of specimen D, where you can find the specimen D in the skin or the body of the domestic fowl. So you can find the quill feather in the wings and also in the tail. The one in the wings is used for flight. The one in tail is used for steering. That is STEERING. Now you may be asked to give the observable features of specimen D, what you have observed in specimen D. So the observable features are: there is presence of inferior umbilicus. As you can see in the diagram, there is also presence of superior umbilicus. There is presence of aftershaft. There is presence of rashes. There is presence of calamus, or otherwise known as Quill. There is presence of barbs. The texture is hard when touched. And then barbs are restricted to certain parts of the quill. As you can see, the barbs are not located in all parts of the quill. Now, you may be asked also to state the adaptive features which enables the organisms to adapt to its environment. So what are the adaptive features that domestic fowl, the quill of domestic fowl possess, which enables it to survive in its environment? Number one is that the vein is flat or broad. Now this enables, it provides surface area for flapping during flight. It provides surface area against desiccation. It's still the same thing. Now, it has centrally placed rashes or shafts, which provide point of attachment to barbs, which also strengthen the feathers. Another adaptive feature is coloration of the quill feather. The coloration of the quill feather enhances courtship display or camouflage. Another feature it possesses is barbs with hooks. These barbs with hooks enables the interlock to form in the vein. Then another feature is that the overlapping arrangement of the quill feather over the body of the birds allows smooth movement and resistant flight. So that is it. And then you may be asked to draw Specimen D. So you really have to know how to draw Specimen D. Make sure you have your magnification, fix your title, as I said in my other video. Specimen E is a longitudinal section of a pineapple fruit. It belongs to the kingdom Plantae, division Magnoliophyta, class Liliopsida, order Bromeliaceae, family Bromeliaceae, genus Ananas, species Commosus. Yes, and the subclass is Zingiberidae. Now, you may be asked to state the observable features of Specimen E. So you are going to say what you see, as we did in our previous specimen. So Specimen E has seeds, or you can say infertile seed, because the seeds are not fertile. They are not fertilized. Another observable feature is presence of crown of leaves, presence of fruit stalk, presence of fruit wall or pericarp, presence of swollen flower stalk that stays in the middle. Then it has rough epicap. The epicap has scars, or the epicap has remains of floral parts. It has oval shape. Now, what is the placentation of specimen E? The placentation is it has axial placentation, and the method of propagation is by sucker. Then what they may likely ask you, what are the modes of dispersal of specimen E? The mode of dispersal is human, just human. Then they may ask you to say or state the reason or explain further. Then you could say that fruits are collected and planted deliberately by man, either for commercial or private use. Now, the life cycle of specimen E is biennial crop. That is, they can survive within the span of 2 years. Other crops that has biennial 2 years lifespan are pepper, ginger, cassava, etc. Then you may be asked also to state the type of fruits or the group of fruits which specimen E belongs. The group of fruits is Soroses. Reason: they develop from several flowers. They are also false fruits. That is, they are formed from ovary and other floral parts. So also try to acquaint yourself with the drawing of specimen E, because you may be asked to give the drawing of specimen E. Specimen F is a transverse section of an orange fruit. It belongs to the kingdom Plantae, division Magnoliophyta, that is flowering plant, class Magnoliopsida, that is a dicot plant. Order is Saphindelis, family Rutaceae, genus Citrus, species Citrus sinensis. Now, what they may also ask you to identify the observable features of specimen F. So, the observable features are: there is presence of fruit wall or pericarp. There is presence of seeds. There is presence of placentation, or you can say axial placentation. It is yellow or green in color. You can choose one, either green or yellow. It has round shape, or it is circle in shape. Now, another one is it has rough epicap. Okay, now you may be asked to give the agricultural classification of specimen F. So specimen F is classified agriculturally as fruit crop. The life cycle is perennial crop. That is, they survive for 3 or more years. Now, the placentation is axial placentation. Now, other fruits that have axial placentation is the pineapple, which we had mentioned, our specimen E. Then other ones are garden egg, pepper, tomato. Now they may ask you to identify specimen F. Specimen F is a berry. The reason is that the endocap is soft and fleshy, and it contains numerous seeds, and you may also be asked to give the mode of dispersal of specimen F. The mode of dispersal is human. Yes, they may ask you to explain further. It could be that the fruit is edible by man, and the fruits are eaten by man, and the seeds are thrown elsewhere to germinate. That could be one reason why, one mode of dispersal of the fruit. Another mode is that man can also collect and plant the fruits deliberately for commercial and private use. Then also you may be asked to draw specimen F. You can see it in the diagram and all parts are shown, so, and labeled correctly. So you can try this also to know that you do well in your exam. Definitely, you will be asked to compare and contrast between specimen E, the pineapple fruit, and specimen F, the orange. So, what are the similarities of both specimens? What do they have in common? Now, their similarities are both have placenta or axial placenta. Both have axial placenta, or you can still say both have placenta. Another one is both are fleshy fruits. Yes, both are fleshy fruits. Another one is both are edible. Another one is both have seeds. Another one is both have fruit wall. If you don't say fruit wall, you can say both have pericarp. Another one is both are dispersed by man. Okay, now what are the differences between the two specimens, pineapple fruit and orange? Now, the differences are, when you are asked to give observable difference, you need to look very carefully so that you can see all the features. So let's try to bring out the observable differences between pineapple fruits and orange. Now, pineapple fruits have infertile seeds, but orange fruits have fertile seeds. Now, pineapple fruit have remains of floral parts. There is an absence of floral parts in an orange fruit. Pineapple fruit has crown of leaves present. Orange fruit, there is absence of crown of leaves. Another difference is that pineapple fruits have swollen inflorescence. Swollen inflorescence is present. Orange, swollen inflorescence is absent. Pineapple fruit, it has rough epicap. It has smooth epicap. Yes, when you compare the epicap of pineapple and orange, you see that of pineapple is rough than that of orange. So we're going to say that the pineapple epicap is rough while that of orange is smooth. Now, pineapple is a perennial fruit. Orange is a perennial fruit. You can still add more if you want, if you can observe better than I do. Specimen G is a raw egg of domestic fowl. If you are asked to classify Specimen G, remember we did something like this when we did our Quill feather. So just go there and copy everything from kingdom to species. It is the same thing. Now, looking at my labeled drawn egg on the screen, you can see it has the shell, the membrane, the germinal thick embryo, the chalaza, and the A space. You may be asked to give functions of some labeled parts. So in raw egg, that's Specimen G, we are going to mention just 3 parts that are present in raw egg. Then we are going to mention the rest when we are doing our boiled egg. So what are those 3 parts that is found in the raw egg which is not found in boiled egg and their functions? The first one is germinal thick. What is the function? It is for fertilization. It is for embryonic development, and it contains the female genetic material. Another name for germinal thick is blastoderm. Another part is chalaza. The chalaza serves as an anchor to the yolk, or it supports the yolk in the center of the egg white, which you can see in the diagram. Now the next one, another function of the chalaza is that it protects the embryo from being damaged during transport or incubation. Another one is the embryo itself. Function, it hatches into egg. Specimen H is a test tube containing 3 ml of water. So what exactly will you be doing with a test tube containing 3 ml of water? You will be asked to conduct an experiment, which is test for protein. Then what exactly will you do? Sometimes your book or your answer sheets may be tabulated into three sections, which contains the test section, the observation section, the inference or the conclusion section. And sometimes you will be asked to provide the experiment, to conduct the experiment, as in say what you do. You're going to write it under the test. Then under observation, you also write what you see. Under inference and conclusion, you also write what you observe. Yes. So let's begin. Under test, the first thing you need to do is you will mix small quantity of egg whites. Small quantity of egg whites, as you can see in the diagram, not the yellow part. You mix the small quantity of the egg whites in specimen H, which is a test tube containing 3 ml of water. That's the first thing you do. Then you add 1 centimeter cube of sodium hydroxide. Yes, of course, you'll be provided with sodium hydroxide and also a dropper so that you can use the dropper to get the sodium hydroxide. So the next thing you do is add 1 centimeter cube of sodium hydroxide and shake the content thoroughly. After that, you also add 1% of copper 2 solution in drops. You will also be provided with copper 2 solution. So you add copper 2 solution to your content or mixture, and then you shake the content thoroughly. So those 3 steps will be under your test column. And under your observation column, you are going to write mixture turns to purple or violet color. Mixture turns to purple is okay. Mixture turns to violet color is still okay. You can take one. Now, the conclusion is that protein is present. The conclusion or the inference is that protein is present. So the name of the test is test for protein or burette test. Specimen J is a longitudinal section of a boiled egg with shell. Now, you may be asked to give the functions of the label parts. You can see from my drawn diagram that the diagram of boiled egg is different from that of raw egg. Now, they are not the same. So, what are the functions? Some of the functions of the label parts or the parts shown in the diagram are the shell membrane, the chorion, the yolk, and the A space. So what are the functions of the label parts? The shell is for protection of the egg. It's for aeration. It gives shape, and it's for gaseous exchange. The shell membrane allows gaseous exchange to be possible. The albumin supplies protein. The yolk provides food for embryonic growth or food for the embryo. The A space allows exchange of gases, and the chorion protects the embryo or the yolk sac. Now, you may be asked to mention the classes of food to which Specimen G belongs. It belongs to the class of food protein and also mineral salts. Example of some of the mineral salts that is derived from Specimen G are calcium, phosphorus, sulfur, manganese, copper. Then just have it at the back of your mind. Let me just add something so that you won't be taken unawares. The importance of protein. Protein, of course, is for growth of young ones. It aids reproduction. It is used in production of hormones, for tissue and cell formation, for production of enzymes, for repair of worn-out tissues and cells. So the classification is the same as that of the raw egg and that of the domestic fowl. So in case you are asked to identify, give reason why you say it is kingdom, it is class, I believe you know what to do. So you may be asked to give the differences between Specimen J, which is a raw egg, and Specimen G, the boiled egg. That's differences between Specimen G and Specimen J. Specimen G is a raw egg and Specimen J is a boiled egg. So they have three striking differences. So what are the differences? Raw egg has germinal discs. That is, germinal disc is present in raw egg. Germinal disc is absent in boiled egg. Embryo is present in raw egg. Embryo is absent in boiled egg. Chalaza is present in raw egg and chalaza is absent in boiled egg. So I'll leave you to bring out the the similarities between the two, the raw egg and the boiled egg. I wish you success in your examination. So if you find this video helpful, please share so that others can benefit. And if you have not watched this video on the screen, how to avoid common mistakes on biology practical exams, please watch before you get into the exam hall. Don't forget to like and subscribe to the channel for more study tips on your exams.